Gernot Zegelbacher, who is a co-chair of the IUCN Conservation Geneticist Specialist Group and a member of the IUCN Task Force on Synthetic Biology and Biodiversity Conservation. Dr. Joachim Mergey, who is the head of the Genetic Diversity Division at the Belgian Research Institute for Nature and Forest. And the two of them will moderate today's chat. Also joining us today as our regional expert is Dr. Uma Ramakrishnan, who is an associate professor at the National Center for Biological Sciences in India. She has been working on molecular ecology and conservation genetics and genomics of Indian birds and mammals for the last 15 years. Now we would kindly ask you to shortly introduce yourselves by saying your name, affiliation, and your role or connection to CBD. So to facilitate this part, I would call country by country in an alphabetical order. So when you hear your country's name, please turn on your mic and introduce yourself shortly. So if you don't mind, we can start. First on the list of participants, I have Belarus. So please, representatives of Belarus, could you please introduce yourselves? We don't have anybody. Okay. Now we have Democratic Republic of the Congo. Then we will move to India. So please, representatives of India, would you kindly shortly introduce yourselves?
Dr. Sandeep?
Yes.
You're muted, I think.
Yeah, Uma. Hi.
Go ahead. You could introduce yourself and maybe, yeah.
I need to introduce myself.
Yes, go ahead.
I'm Dr. S.K. Gupta, Sandeep Gupta, working in Wildlife Institute of India as Nodal Officer, Wildlife Forensic Cell and Conservation Genetic Cell. And I have about 18 years of experience in this field, so enjoying working in this field and hope to listen good things in this seminar. Thank you.
Thank you so much. Do we have anybody else from India attending the webinar? Okay, maybe we can move on and we'll see if people will join us in the meantime. So now we have Indonesia.
Hello, good afternoon, everybody.
Hello.
I'm Gona Semiadi. I'm from Indonesian Institute for Sciences, and I'm been assigned as the national focal point for Substa CBD. And thank you very much for allowing us to get involved in this webinar and seeing toward for the discussions. And we do have here several members from Indonesia, but it's quite many numbers. So I believe that will be fine represented by myself. Thank you.
Welcome. Thank you so much. Now we have Iran. Maybe move to Malaysia.
Hello, I'm Roslisa from Malaysia Agriculture Research Institute. I'm just joining, so happy to meet you all. Hope for to get.
Thank you so much.
Yep.
Thank you so much for joining. Yes. Do we have a representative of Myanmar?
Hello.
Hello.
Yes. We are the Myanmar Seed Bank. Nice to meet all of you. Yeah.
You're welcome.
Yes.
Okay.
Thank you so
Okay,
much.
see you.
Thank you so much
Yeah.
for being with us.
Okay. Yeah. Yeah. Thank you.
Do we have a representative of Nepal?
Just we have the representative of the Myanmar State Bank.
Okay.
Yes. Yes. So I'm the documentation and database manager. Yes, I'm the Miss Omar. Yeah. Senior research. Senior research assistant. Yeah.
Thank you so much.
Yeah.
So do we have a New Zealand? Oh,
But I introduced to the our boss.
Also
yes.
another one from Myanmar.
thank you so much.
Hello.
Welcome.
Yes.
Thank you for joining.
My name is Ante. Yes, I am working for the Department of Agricultural Research as well as the Gene Bank. Yeah.
Wonderful.
Yes.
Thank you. Thank you.
Thank you.
Good afternoon. Hello.
Hi.
Yeah, I'm
Yes, go
Timade, assistant. Yeah, I'm from Myanmar. I'm Timade. I'm assistant director from Environmental Conservation Department. I'm working for Natural Resources Division. I'm an assistant for National Focal Point on NACA Protocol. Nice to meet you all. Thank you for joining this webinar.
Thank you so much for being with us today. Thank you so much. So we now move to New Zealand.
Yes, hello.
Hello.
I'm Dr. Tim Strabela from the Environmental Protection Authority in Wellington, New Zealand. And I'm New Zealand's national focal point for the Biosafety Clearinghouse.
Wonderful. Thank you so much. So now we move to Philippines. If not, let's go to Republic of Korea. No. Do we have somebody from Singapore?
Yes, good afternoon. This is Wendy
Hello.
from the National Parks Board of Singapore, and I'm the CBD as well as the National Focal Point. Thank you.
Wonderful. Thank you so much for being with us. So if this is it, so we move to Togo. Do we have a representative Togo with us today? Okay, so since we had a couple of participants joining while we were going through the introduction, I would like to call those who joined us in the meantime, if they would kindly take the stand and represent themselves, saying a name, affiliation, and the connection to CBD. So please, all of those who didn't introduce themselves, would you please do it now? Hey, just turn your mic on. You're muted.
There seems to be a problem with your mic,
Your mic, yes.
Madam Kumar Katka. If you want to just put your self-presentation in the chat, would be fine as well.
Yes.
And then you might solve the problem with your mic.
Mm-hmm.
Someone just joined.
I think from Belarus just joined.
Yeah.
So I think we have a representative of Belarus now in the webinar. So if you would kindly turn on your mic and say your name, affiliation, and the connection to CBD, please. Hi.
Needs probably a moment. It's now it's connected.
Yes. So please, the representative of Belarus that just joined, could you please introduce yourselves? just saying your name, affiliation, and the connection to CBD.
Hi, can you hear me?
Hi.
My name is Galina Mozgova. I'm representing the Institute of Genetics and Cytology of the National Academy of Sciences of Belarus, and I'm DCH National Focal Point. Also, I'm participating in many groups at the CBD on LMO risk assessment, on socioeconomic considerations, on genetic resources, and many others.
Wonderful. Thank you so much.
Thank you.
Do we have maybe somebody else that didn't introduce themselves that would do it now?
Hi, I'm Ruliana Susanti from Indonesia. I work in the Center for Biology, Indonesian Institute of Sciences, the national focal point for SAPSTA.
Well, thank you so much.
Nice to meet you all.
Nice to meet you as well.
Yes, my name is Yuyu Purba. I'm also from Research Center for Biology, Indonesian Institute of Sciences. I'm a researcher in plant genetics and breeding of bananas. Thank you. Nice meeting you all.
Nice to meet you. Thank you so much. Anybody else, maybe? Maybe that's it for now. So thank you so much for being with us today and introducing yourselves. It is wonderful to see this diverse group of representatives from different countries. It is wonderful. Now I would like to call Dr. Vernesi to introduce all of you to our cost action that is called G-Bike. So please, Dr. Vernesi, go ahead with your presentation.
Thank you. Okay. Can you see it?
Yes.
It's fine? Yes. Okay, good.
Yes, it's fine.
Okay. Again, good afternoon to all of you. Let me first introduce a brief overview of our cost action. So what is a cost action? It is a specific program funded by European Union for networking activities. So basically, they give you funding for promoting a specific networking like workshop, training school, short-term certificate mission, which are short stay of researcher in different countries in a certain subject. So as you can see, our acronym stands for Genomic Biodiversity Knowledge for Resilient Ecosystems. You can see listed here all the people being in charge of specific role, like working group coordinator or science communication manager, like Dr. Nevena Velikovic, who is attending today's webinar. Okay, which is the specific challenge of G-Bike? We would like to establish the use of genomic data as a standard tool for monitoring and managing wild and exceed populations of plants and animals. So all species, not just the one with economic importance, but really all the species, especially in our view, species from the wildlife. And of course, we would like to implement the best practices for maximizing species potential for adaptation through promoting the measuring and monitoring of genetic diversity. And of course, we would like to finally introduce these practices in the new European Union initiative, like the European Biodiversity Strategy for 2030, not just 2020, of course, and other directive like Habitat Directive and Natura 2000 networking sites. Why the idea of the bike? So we were thinking from the very beginning, but it would be extremely important to have the two wheels, one represented, as you see here, from the right side by practitioners, and on the left hand by scientists. Insofar, probably we experience a kind of decoupling of these two wheels. So we're thinking thinking about connecting the two wheels with the frame of the bike in order to really have a proper movement. I would like to remind to all of you that our current project, G-Bike, stems from a previous successful experience, again funded within a European Union granting scheme, but was called a Congress, and probably some of you are aware of. As I said, the main tools for promoting our networking activities are training school offered to people at various levels of knowledge, so practitioners, scientists, young researchers, workshops, short-term scientific missions, which are short stay from 5 days to 90 days in a certain lab, not for doing research, but for promoting advancement of some ideas, and webinars like the one we are holding right now today. Yeah, genetic variation, we know it's the key to adapting to environmental change. So genetic diversity is expressed at DNA level, and it's the basis of any biological difference, the one you can find between species, but also the one you can find among individuals of the same species. We know now from a long time that some individuals are better suited to survive and reproduce in a certain environment due to their genetic diversity. I would like to remind all of you, please, to mute your mic while you are not speaking. Otherwise, we receive a background noise, which is a little bit disturbing while doing presentation. Thank you. Okay, so genetic diversity in the ultimate way, it's something that increases our probability of survive in the environment. So giving the possibility at the ecosystem level to have resilience, and of course to give ecosystem the opportunity to produce the most relevant services. And we also know now since long time that when population becomes small and isolated, they rapidly lose genetic diversity. And so we need to enlarge and connect the different populations in order to let them retain the capacity to adapt to environmental change. So what we propose that measuring and monitoring of genetic diversity would be introduced in any new policy regarding biodiversity, preservation and conservation, like for instance, the current discussion of the conservation of biological diversity, so the global biodiversity framework post-2020. That's the reason why we are holding this webinar today. And we know that ultimately species health depends on genetic diversity, and but the introduction of the monitoring and measuring genetic diversity is going to really significantly improve the management of biodiversity and natural resources. And finally, I would like to stress the fact that it's now that we need to act, especially now that the different meetings from SEPSA to open-ended working group are taking place. So the reason why we are so committed to this strategy is really for sake of introducing this concept to you and hopefully make them understandable, so that afterwards you might promote the introduction of this measure, these indicators into the new CBD policy. That's all from my side. I stop here, and of course we can discuss later on about specific questions on this part. But I guess for the moment it's enough, and thanks again for your attention.
Thank you, Dr. Veronesi, for the introduction of our cost action. So as you all know, the conservation of biodiversity is a common concern of the humankind in general. So the Convention on Biological Diversity covers biodiversity at all levels: ecosystems, species, and genes. And as you saw, the gene level is the focus of today's webinar. So because the overall objective of CBD is to encourage actions leading to a sustainable future, we highly value your participation in today's webinar. because you represent your countries in the meetings, you identify experts to participate in the processes, you collaborate with other stakeholders such as G-Bike and other focal national points, and you monitor, you promote, and you facilitate the implementation of the convention in your respective countries. So once again, thank you so much for being with us today and listening to the presentations. So now we will move to the main part of the webinar, which is the presentations from our experts. So we would like to encourage you today to interact with our experts as much as possible. And there are two ways of doing so. You can either write your questions and comments in the chat during presentations, or you can use options raise a hand in the participants tab to post a question in person during the question and answer section following the presentations. So once again, we will kindly ask you to keep your microphones muted during the presentation, so to avoid the sound interferences. And before we start, we would like to remind you that a certificate of participation will be provided for all attendees of today's webinar. So our first expert today is Dr. Linda Laikre from Stockholm University. Professor Laikre is a population geneticist with a strong interest in conservation biology. She is the deputy head of zoology department, head of division of population genetics, and director of studies in population genetics. The topic of her talk today is why genetic diversity is important and fundamental concepts, including effective size population, or NE. So Dr. Laikle, please go ahead with your presentation.
Thank you very much for the introduction, Belma. So I will share my screen. And I'd like to say good morning to everybody, of course. I'm really happy that you are here. And I'm starting off by showing you an olive, a nutritious fruit, a part of biodiversity that many of us like. And it's also a good way to symbolize the relationship between census size, the actual number of individuals in a population, and how the population behaves genetically. So census size is often abbreviated as NC, and effective population size, which is how the population behaves genetically, is symbolized by NE. And this graph shows them that a population can behave genetically as much smaller than what census size indicate. And of course, this is very important in biodiversity conservation, because it's this size that determines the rate of genetic change. So the effective population size, NE, is usually much smaller than what census size is. And it is the effective population size that determines the rate at which we lose genetic diversity. And I'm going to talk a little bit about this concept because it relates to one of the indicators that we propose to be used within the CBD context in the next biodiversity framework post-2020. So going back very basic to biodiversity, we know ecosystems are systems of different populations of species that interact with non-living components of the environment and creating biological systems with specific functions. And of course, species we know are key to ecosystem functioning, because if we lose a specific species in an ecosystem, that can erode the whole system and make it collapse. And similarly to this importance of species biodiversity for ecosystem function, genetic diversity within species can have similar effects. So if we have a species that belongs to an ecosystem, it is in fact built up of a lot of diversity within the species. So we can have genetic differences between populations of the same species. And I symbolize genetic diversity with colors. So here we have, in this population of the species, we have a lot of orange alleles, gene variants of a specific type that do not occur in this population, for instance, which is the same species, but another population. And this diversity here can have equal effects on an ecosystem. So if we lose a genetically distinct population in an ecosystem, that can also cause the ecosystem to lose its function. So genetic diversity is extremely important, and currently it's not included in the monitoring of biodiversity. So all this variation here below the species level that consists of genetic diversity within and between different populations, that is built up of genetic variation at the DNA level. So we have genes that can occur in different variants, which we call alleles. And again, I symbolize genetic diversity with colors here. So all of this diversity, which is the tools for continued evolution, is currently not included as in indicator work within the CBD. And that's what we think is necessary to change for the coming framework, because this diversity is the basis for everything else. Without diversity at this level, we will lose the potential of species to be able to adapt and evolve into the future. So what is at focus for our presentations today is naturally occurring genetic variation within and between populations of species. Such variation is necessary for populations and for species and for ecosystem to survive and keep their resilience. And we are not really discussing genetically modified organisms, GMO, or other kinds of genetic manipulations. So we are focusing on naturally occurring genetic variation. And we know from many decades of genetic research that high genetic diversity, again, I symbolize genetic variation with colors. If you have a population with a lot of gene variants, high genetic diversity, such populations typically have high adaptive capacity and a good potential for surviving in the long term. It has high resilience. And in contrast, we know from many empirical studies that low genetic diversity, so here we don't have so much genetic variation, such situations are typically coupled with low adaptive capacity, weak potential for long-term survival, problems with inbreeding effects, and low resilience. And there are many examples from the scientific literature on these things. And just to mention one, we have the Atlantic killifish, which is a really small fish that occur along the North American coastline. And researchers have reported that genetic diversity have allowed populations of these fish to adapt to extremely toxic environments. So environments that were originally toxic for this species, due to genetic variation, some populations have been able to adapt to those kinds of situations. So that's an example of how a lot of genetic variation can aid populations to be able to adapt to new changes in the environment. And as an example of the problems with low genetic diversity, again from North America, we have the wolves on the Isle Royale, which is an island in Lake Michigan, where wolf population have been occurring for many decades, but they have been isolated and the inbreeding has become extremely high, and that has caused the populations to completely collapse. So only two individuals have remained in this population. They try to save it now, but inbreeding caused the population to collapse completely. And there are many other examples of that, and we have some of them in our policy brief that you can find on a GBike website, and I think you will get the link into the chat soon. So genetic diversity is extremely important to conserve. So what kind of processes can lead to loss of genetic diversity? We have some general processes that can result in such loss. For instance, if genetically distinct populations go extinct, then that means we will lose genetic diversity. If populations become really small, we have reduced population size, that will also cause loss of genetic diversity. If we induce selection pressures, for instance, through harvesting wild populations. So if we go out and, for instance, catch fish, and we like large fish, the red fish here, and if this size, body size, is to some degree determined by genetic variation, that means genes that cause this large body size will be removed from the population, and we can lose such genetic variation in the population. And the fourth type of loss of genetic diversity can occur through release of non-native genes. So if we have a natural population here, for instance, that receives individuals from hatchery breeding, for instance, I have fish here as symbol. So let's assume we have a hatchery breeding of a specific species and then release it into nature. The native gene pool can become lost if this release is very extensive. So these are 4 categories that results in loss of genetic diversity. And our indicators are aimed to deal with these losses. So we have an indicator 2 that you will hear more about soon that focuses on maintaining populations. So this type of loss will be detected by our indicator 2. Our indicator 1, which focuses on genetically effective population size, will be able to detect situations where reduced population size result in loss of genetic diversity. And our indicator 1 and indicator 3 will be able to deal with these kinds of situations. And our indicator 3 will also be possible to use for detecting loss that is caused by genetic homogenization through releases. So key questions for monitoring is, of course, how fast does my population lose genetic diversity? And is my population large enough to maintain sufficient levels of genetic diversity? So these are key questions that we need to answer for our populations. And effective population size can provide an answer to these questions. It's a quite complex concept, but our indicator 1 we have designed to simplify this to be able to use it in CBD purposes. So just to explain a little bit about what genetically effective population size is, the definition is that it's the size of an ideal population with the same rate of loss of genetic diversity as the real population. And that's a bit complicated. So a little bit simpler explanation is that it's the number of individuals that reproduce and also taking into account how much those individuals contribute to the next generation. And effective size determines how fast the population lose genetic diversity. So as effective size increase, the rate of loss will decrease. And many factors affect the genetically effective population size. And just to mention one of these, is a sex ratio. So there are characteristics that makes the real population not ideal. And one such thing could be the sex ratio. Under ideal condition, the sex ratio is equal. But that's often not the case in natural populations. So here is an example of a non-equal sex ratio. So we have an NC, a census size, number of individuals is 100, but the sex ratio is very skewed. So we only have 1 male and 99 females. And in contrast here, we have an equal sex ratio. So this is an ideal condition. The census size is 4 and we have 2 males and 2 females. And the relationship of effective population size taking sex ratio into account looks like this. So if we want to compute how large NE is, we take 4, multiply it by number of males, which in this case is 1, multiplied by number of females, in this case 99, and then we divide it with the census size, number of males plus number of females. So using this equation, we find that the effective population size in this case here is approximately 4, and it's the same in this population. So here we have the same effective population size. The rate of loss is the same in these two populations, even though their census size is very different. And this is because this skewed sex ratio here results in a real strong reduction of effective population size. And there are other demographic processes that also can affect effective population size. And one such parameter is individuals. In an ideal situation, individuals are equally likely to contribute offspring to the next generation. And we will not go into details here, but just looking at, let's assume this is our population. We have these individuals with lots of genetic variation, again symbolized with colors here. Let's assume that only 2 of these individuals reproduce and produce offspring to the next generation. That means only the genes from these individuals will be transferred to the next generation, and we will lose the genetic variation of those that did not reproduce. So if few adult individuals reproduce and contribute offspring, that will result in loss of genetic diversity, that it goes faster, and that the effective population size is reduced. And there are many other demographic factors that can also affect genetically effective population size. So most real populations do not fulfill ideal conditions, and that is why the effective size is much smaller than census size typically. And so how large effective size do you need to be safe, to be sure that you conserve genetic diversity? There is a scientifically well accepted rule of thumb that says that if effective size is equal to or larger than 500, that's large enough to maintain sufficient levels of genetic variation to be able to make sure that adaptation can occur. So we suggest to use this rule of thumb as a limit for our indicator 1. So we propose for indicator 1 for CBD purposes, the number of populations within species which has an effective size, NE, above 500 compared to those that have a low number below 500. So we suggest to monitor effective population size of populations. And then, so how can we assess effective size? We need to know if our population has effective size 500 or if it's below 500. And there are several ways to obtain an estimate of effective population size. One thing is if you have demographic data of the type that we just looked at, how much individuals are reproducing, how much offspring they contribute, and the sex ratio, and so on. But if you don't have that, you can also use simulation programs that need some basic demographic data, but not so detailed as for these equations. A third way is to use genetic data, go out and collect tissue and perform some sort of molecular genetic analysis or a genomic analysis, whole genome sequencing. And so there are different ways to use molecular data. Some methods need 2 points in time. So you need to collect tissue from 2 different time periods from the same population. And there are other methods which only needs 1 point in time to estimate effective size from genetic data. And there are many such estimates available from the scientific literature because this has been done over many decades in conservation genetic research. But if we do not have the possibility to do any of these, we don't know any demographic data, we don't have the potential to do simulations, and we can't do genetic studies, then we can use census size as a proxy. So, and that is what we suggest for our indicator 1. A proxy for effective size in the absence of detailed data is to use 10% of census size as an estimate of effective population size. And this proxy, the relationship between NE and NC being 10% or 0.1, we think is a good proxy because several scientific reviews have indicated that mean or median values over many species, many populations is approximately 10% or 0.1. And in some studies where very detailed genetic monitoring have been carried out over generations, this estimate has also been found around 0.1 or around 10%. So assuming that NE is approximately 10% of NC, we think is a good way forward for this indicator. And to obtain our NE 500 then, we need a census size of 5,000. And we think that this can be used in cases where detailed data is not available. And by that, I would like to thank you for your attention.
Thank you so much, Dr. Laikre, for your wonderful presentation. And thank you for keeping with our schedule. Therefore, we have plenty of time to start a discussion now, if you don't mind. Now it is the time to post questions, your concerns or comments. You can either post it in the chat box if it's easier for you, or you can post questions in person. So I propose that we start with a question that we got during the presentation of Dr. Laikre. That was posted from Mrs. Yayuli. I apologize if I pronounce last names incorrectly. So the question is there any different calculation for effective population size for animal and plant population?
Yes, thank you very much. This is a good question. Yes, there are many different calculations of effective size from a whole variety of plants and a whole variety of animals. And we have a scientific paper on this topic where we have also summarized all this information. There are several reviews available on this which have compiled and lists all the populations for which we have effective population size estimates. So I think we can put the link to this paper in the chat here, so you can go have a look at it, because there are a lot of data available, and there is a lot of variation in effective size and the ratio of effective size to census size depending on the organisms. So as we have more details, we can do better genetic monitoring. But we think that for CBD purposes, it's important to have a simple indicator that can be used worldwide. And then it's possible for different countries to go into detail. I mean, for instance, in Sweden, we are right now starting genetic monitoring using genetic data. And when we have such data, we can go into greater detail on the different populations that we are studying.
Thank you, Dr. Laikre. So, yes, so we have a comment from Joachim in the chat. So for most plants, this is pretty similar and the same principles hold. Yes, thank you so much for your comment. So please, we would kindly ask our participants to make comments, pose further questions, or maybe your concerns and doubts. Yes, we have another comment in the chat section. So it says it's from Mr. Sanyadi. And the question is, effective population size in isolated island is a problem for such an archipelago country like Indonesia. Some islands harbor an endemic species level and others as subspecies with a population only between 300 and 500, stable for almost 20 years. Will this be becoming a problem?
Yeah, please, Joachim.
Well, it depends on the species, but 20 years in evolutionary timescales is very short. So if it was a much larger population before that, it could well come into troubles because of loss of genetic diversity. So for many species, this will in the end, and many populations that are this small, this might very well become problematic in the end.
That's exactly what happened in this example of wolves on Isle Royal. That was also an island, a quite small island. And it was often used as an example that we didn't have genetic problems because this population was doing fine for 6 decades, so 60 years before the genetic collapse. So that's very important aspects of genetic diversity. We expect things to take some time before we see the negative effects of loss of genetic diversity.
If I could just add, add a point, Linda, thanks for bringing up the aspect of time, because this is something which, if you've talked a lot with managers, you'll find is a big bone of contention, because often managers are thinking in a very short time scale, whereas a lot of the processes we are referring to are longer time scales. So this is one of the first very important messages to communicate. I think it's really important for us to all internalize this evolution, these long time scales. Thank you.
Thank you, Dr. Ramakrishnan. And we had another question posted in chat from Dr. Gupta, who said, what is acceptability of introducing genetic diversity in a natural population? How to deal with persons who are resilient in doing so?
Well, I mean, this is also a quite difficult question because it's difficult to foresee what kind of genetic diversity is needed in a specific case. Of course, it's very important to try to have populations non-isolated to maintain the connectivity between populations. That is a really important aspect and something that needs to be taken into consideration. So I think that's the main thing to try to save genetic diversity. Make sure that you maintain genetic connectivity among your populations and perhaps assisted gene flow between populations if that becomes difficult. Please, Joachim, perhaps.
Yes, I would like to add to this. It's important to try to restore the process. So we can, in some cases, add genetic diversity as a short-term relief for acute problems. But if you're not resolving the initial problem that caused this loss of diversity and this loss of connectivity, we're just keeping a zoo eventually. So it can be used indeed to solve acute problems in genetic diversity. And there are guidelines from the International Union for the Conservation of Nature for this. Different countries also have guidelines targeted at their own situation. And from the genetic perspective, it is generally thought to be more often beneficial to add genetic diversity than the opposite. So the reasons not to do so would be you could get outbreeding depression, which is a loss of fitness because your populations that you are mixing are too different and adapted to different environments. So this is a risk. But there is a decision tree for when to consider that outbreeding depression is too high a risk. And this is something that we have not considered in our guidelines, but which are available in the international literature and which we could provide as well.
Dr. Ramakrishnan, you also had something to comment?
Yeah, I just add that in many parts of Asia, with ongoing incredible land use change, these factors are very important. And I think that going ahead, robust criteria, as suggested, an internationally accepted criteria for assisted gene flow will be a really important topic to think about. Thank you.
Thank you so much. And we also had a comment from Dr. Degelbacher in chat saying that in case of genetic rescue, there is obviously also an IUCN guidelines for reintroduction, as Joachim mentioned. So we can maybe move to another question posted by Dr. Strebela from New Zealand. It says, do you see a major role for genomics-assisted breeding in improving effective population size in populations as in being done for kakapo in New Zealand?
Yes, I mean, I think this can be of help, of course, in dramatic situations such as the one that you bring up. But we are trying to introduce indicators and guidelines and work to try to avoid such situations. So we like to maintain populations at sizes that are large so that we avoid ending up in that kind of situations that you need those extreme measurements. So that's why we think it's really important to include goals, targets, and indicators that try to avoid, that maintains populations at large enough population sizes with large enough connectivity, so we don't end up in really poor genetic situations. Joachim, did you want to add something? Okay.
And we also had a comment in chat by Dr. Hertz regarding the question on the islands. And she said for plants on islands, there may be a connection through some level of gene flow, such pollination seed dispersal among islands. So the situation may not be as dire as for some animals that may be more isolated on islands. So thank you, Miriam, for your comment. Yes. We also have a comment from Dr. Sanyadi. IUCN concept is based on natural distribution, whilst our management system as a country level. So some species is considered as an outpopulation by the scientist due to its strict distribution and not by the management. This always gives problem in further coordination.
Yes, that's true. I mean, that's a general thing that we need to have at some point collaborations between countries to be able to make sure that we maintain genetic diversity of populations. I think Per will bring up an example of that later on today.
And this is one of the roles of GBIKE as an action to bring this community of scientists and managers closer together, working on the same goal. Yes. Do we have maybe another question in this section, or would you prefer to move to another presentation and then we can continue discussion afterwards? So maybe it would be time to move along. Yes. So our next presentation will be by Dr. Sean Hoban from Morton Arboretum in the United States of America. Dr. Hoban works closely, among others, with the Society for Conservation Biology and the International Union for Conservation of Nature, IUCN. The topic of his talk is towards genetic diversity indicators, what they are, how they support goals and targets, and their use for conservation policy support. However, due to the time difference, Dr. Hoban could not be with us in person today, but he kindly provided us with the recordings of his lecture. However, to answer your questions regarding his presentation and to facilitate your understanding of that topic, with us is our expert on plant ecological population genetics, Dr. Miriam Hertz, from the French National Institute for Agriculture, Food and Environment. So now let's listen to the presentation of Dr. Hoban.
Hello, everyone. My name is Sean Hoban. I work at Morton Arboretum, which is a
it's working for everyone.
Botanic garden and plant science research institute outside of Chicago, Illinois, United States. I'll be talking to you about recently proposed indicators that can help improve the post-2020 CBD Global Biodiversity Framework, as well as goals and targets connected to those indicators. Everything I'll talk about today has been published in these two articles. This one, Biological Conservation, is open access, free to read for anyone. And as I'll mention at the end, we also have a short 2-page summary, very accessible for people. The reason we're holding these webinars is because that in the past, CBD has relatively neglected genetic diversity in its commitments through vague targets as well as weak indicators. We propose in our article and in this webinar several improvements to how genetic diversity can be considered by the CBD. Those improvements include an improved goal with much more clarity on genetic diversity, an improved target, specific actions that can help genetic diversity, and 3 specific pragmatic indicators that do not necessarily need genetic data, at least these do not. So these indicators, the action target, and the goal are very well connected. And an important point is that we aim for the post-2020 CBD framework to protect genetic diversity within all species. So this is a diagram of a food web on the left. Species are very interconnected, and the loss of genetic diversity in one species can affect the resilience and the diversity and the survival of other species in that food web. So it's very important that we can conserve the genetic diversity of all species, not just domesticated species like livestock and crops. In our proposed modifications to the CBD post-2020 framework, we also suggest that our long-term goal needs to be restoring the conditions ensuring adaptation. So not just do we need to halt the loss of genetic diversity, but we need to do restoration, connectivity among populations and large population sizes, as well as monitoring to ensure that adaptation can take place for the long term. Our three indicators, they were shown on the previous slide, but they boil down to this. Populations need to be large enough so genetic diversity is not lost due to random processes. Populations themselves cannot go extinct because each population can contain unique adaptations to help a species survive. And we need to monitor species and populations using demographic methods, but also genetic methods, some number of species and populations, so we can get a sense of overall trends at a country level. Here's what I'll talk about today. I'll touch briefly on past CBD indicators, their strengths and weaknesses. I'll talk about these new indicators, connections to policy, actions, data sources, and I'll come back to that goal and target. As many of you probably know, the CBD had a 2010 and a 2020 or Aichi target on genetic diversity. These both focused on maintaining genetic diversity, and in this case also associated indigenous and local knowledge. In both cases, these targets focused on either valuable species or domesticated animals, their wild relatives, and some secondarily socioeconomically and other valuable species, including culturally valuable. But the main focus has been on cultivated plants and animals, specifically crops and livestock, the genetic diversity within those species. So to measure progress on these targets, CBD has had indicators in the past. These are the 4 indicators that were presented in the kind of 0 draft and that have been used for some time, for the most part. What do these actually represent? The first one primarily represents the size, usually in number of species or number of accessions of seed banks at national levels. This one represents rare or indigenous or local breeds of animal, usually livestock, and how close those are to extinction. I should note that breeds are similar to populations. They're usually adapted in some way and have specific traits. The Red List Index has been used to report on genetic diversity, but it's actually not well connected to genetic diversity. It's closeness of a species to extinction. And then the comprehensiveness of conservation was recently proposed. It measures the amount of a species' geographic range that has been conserved. Overall, these indicators are not great for monitoring genetic diversity of wild species, either because they ignore wild species entirely or because they don't actually track closely to conserving genetic diversity, with the exception of this 4th one. So we recommend 3 pragmatic indicators that focus on 3 different areas of genetic diversity conservation. The number of populations that are large enough, NE above 500. Linda explained a bit about what NE is, and we're happy to answer questions, but the number of populations with an NE large enough, typically above 500, to prevent genetic diversity loss, preventing the loss of populations, and the number of species and populations where genetic diversity is actually monitored, preferably using DNA. The first indicator, number of populations above 500 NE, this, as Linda explained, will prevent the random loss of genetic diversity that happens in small populations. This is the primary mechanism that will prevent genetic erosion within populations and ultimately at the species level. Interestingly, the CBD has recognized the importance of effective population size for a long time because that is the basis of declaring a breed as threatened. So NE is used currently to declare breeds as threatened. And like I said, breeds are very similar to populations. So actually this new indicator should not be difficult for the CBD framework. focusing on threatened populations instead of threatened breeds. Okay, the clear policy lever or action that can be taken is to make sure populations are large. The data sources that countries can use to report on this include the Living Planet Index or IUCN assessments, not the actual ranking of threat, but the data contained within those databases, as well as genetic data or surveys at the national level. And Per, in the next talk, will give some other examples of data sources and how these this indicator can be reported on. So he will give some specific examples. The second indicator, proportion of populations maintained, this is slightly different. This focuses on unique adaptations. So species usually occur across quite a few habitats and geographic range and environments, and we need to maintain all those populations to keep the adaptations available for the future, for future option. Interestingly, again, the CBD has recognized the importance of this kind of data about biodiversity with the new comprehensiveness index that I mentioned. That's the fourth existing indicator. Okay, the clear policy lever or action that can be taken to increase the status of this indicator is to stop populations from being lost. Countries can use similar data to the last one, but also including potentially GBIF, the Global Biodiversity Information Facility, or other large databases of species occurrences, including potentially citizen science data. So citizen scientists and non-experts can take a role, including indigenous and local peoples, can take a role in providing data on here. And again, Per will give some examples in the next talk for this. The number of species and populations where genetic diversity is monitored, preferably using DNA-based methods. Ultimately, we need knowledge on how genetic diversity is changing. The previous two indicators are proxies. In many cases, they will be suitable, but we still need knowledge of genetic diversity status and change, not only for reporting, but for managing species genetic diversity. So interventions like translocations, building habitat corridors, starting ex situ breeding programs, these all depend on knowing the status of genetic diversity in a species. So we do need some number of species, depending on the country's capacity, to be monitored using DNA-based methods to understand inbreeding and other genetic issues. The CBD has recognized the importance of knowledge and observations for management, and in the previous AHE Target 19. So this new indicator on genetic diversity observations should also not be too hard to fit into the CBD framework. The policy lever that a country can take to improve the status of this indicator is to commit to long-term monitoring, funding of applied research, and also communicating results. So connections between scientists, managers, and policymakers, and between scientists and the public also need to be funded. The data source can be measured in terms of number of reports, number of communication mechanisms, open data sets that are submitted to things like GenBank and Dryad, other publications, that kind of thing, or number of formal monitoring programs. Like I said, these indicators need to be connected to a goal and a target. Ideally, goals, targets, and indicators should be very well connected for every indicator, goal, and target. So we foresee a long-term goal for genetic diversity, that adaptive potential is secured and restored, and that long-term adaptive processes can help maintain genetic diversity within populations and species. By 2030, we think we can at least halt current losses of genetic diversity and develop and initiate strategies which will get to this 2050 goal. An action target falls between the goal and the indicators. So an action target should focus on the specific actions that can be taken to improve the status of indicators and then achieve the goal. We suggest the actions needed are very clear. Very clear. Keep large populations, ideally as many populations as possible above an effective size of 500. I should note these are ambitious targets, goals, and indicators. In some cases, they will not be achieved for all species, but I think that's okay. We need to give a realistic picture of biodiversity status and change. And sometimes that means that for some situations or species or ecosystems, we aren't reaching our targets. But that's okay. It gives us something to improve upon. Okay, the second action is, like I said, halt the loss of populations. And the third action is to make sure this monitoring happens. Within key species, we understand that all of these indicators would not be for every species in a country. So some representative set of species. So anyways, these are three things that should be in a genetic diversity target. And it should include ex situ and in situ. An example wording is given here, but we could work on that precise wording. There are challenges to these indicators. In some cases, countries or programs may not have good data for many species. Of course, that will be the case. But we are working on identifying good data sources. Populations, loss of populations and low NE doesn't have a linear relationship with loss of genetic diversity. And so we do need to work more on identifying thresholds that are really, truly important, that are kind of critical red flags for changes in the status of these indicators. And then, of course, the third indicator looks at monitoring. It's not necessarily related to the status or the outcome of genetic diversity. It measures the effort being applied. So that's an important caveat. Nonetheless, these indicators are smart, they are specific, quantitative, they are measurable. There's data available for many countries from a variety of sources. They are achievable or feasible. We can take action and make them happen. They are relevant. They are connected to key concepts in genetic diversity conservation. They are as close as we can get without getting too technical or genomic to actually measuring genetic diversity conservation. And they are time-bound. We have baseline data and we have goals that are time-bound. Our group, representatives of IUCN, GEOBON, the Society for Conservation Biology, and this organization called GBIKE, along with hundreds of conservation geneticists and partners in governments and agencies around the world, are working to improve these indicators. Our next step is to assess how much data is available per country, and we have several countries we are starting to work with on case studies. The next step is to actually calculate the indicators at a country level. In the next talk, Per will give some small examples, 5 short examples, but ideally we'd want to do that for 100 or more species at a country level. And then to actually deploy the indicators with scientific and technical advice, a continuation of this webinar series as other handbooks and guidance to help countries use these indicators. That is soon to come. Our group consists of many diverse scientists from around the world, dozens of countries, from dozens of organizations, not just academia, so we're not just research scientists. Many of us work in practice, and we have close partnerships with important biodiversity institutes around the world. And this helps us to understand practically what are the data availabilities, limitations, and the needs that countries have. And we welcome you into our partnership and to give us feedback and suggestions. All of this can be summarized in a short 2-page document, which is freely available on the web. And I think we've sent it to all of the participants for a short summary for policymakers. We have a new paper coming out which further underscores the need for new genetic diversity indicators. Our paper, which is in review, talks about how countries still are mostly focusing on agricultural genetic diversity and are not reporting much on genetic monitoring or specifics on genetic diversity change. So we really do need these indicators so that we can improve our national reports in the future. So I hope I've convinced you, and the other talks have convinced you, that our indicators, as well as associated goals and targets, are relevant, they are sensitive to genetic change, they are scalable, they are countable, and that there is some data available. We would first like your feedback to improve these. We would then appreciate your support through the upcoming Substa and open-ended working group and other interactions with CBD to ensure that genetic diversity indicators are included in a post-2020 framework, and that we have a stronger goal and target focused on genetic diversity. And we can work together through upcoming CBD events and online feedback. We can help provide text for suggested improvements or interventions at events. We really look forward to working together with you to conserve genetic diversity for a more resilient planet and society in the future. Thank you. I'm sorry I won't be here to answer questions, but now is the time for questions. Thank you very much.
We kindly thank Dr. Hoban for providing us with his presentation. As he suggested, it is now time for questions. In the meantime, during the presentation, we received several questions. First came from Mrs. Wendy Yap, who is Singapore's national focal point at CBD. She posted two excellent and relevant questions. The first regards the selection of species for monitoring, but maybe that one we can save for after Per's presentation, because the next presentation will relate to that question. But the second one is, how data intensive are these indicators at the national level?
Perhaps I can say a small word on these things. So actually we are here several people from G-Bike who are in charge of different aspects of genetics in this action. I am myself the leader of the working group 3 on genomics and ecosystem services. And then we have, of course, Joachim and Gernot here, who are population geneticists and very strongly involved in their research on aspects of genetics. And both of them are leading initiatives into genetic monitoring. So I think we will be mostly the trio of us trying to answer your questions you may have. So perhaps to nevertheless say a small word on the selection of species. Joachim has already mentioned here in the chat that it will be important to cover different species, a selection of species that have different traits. And that is quite an important aspect. You should go into trying to look at small species, large species that have different ways of reproducing, numbers of offspring, perhaps also look into different levels of the trophic network, as Sean was showing in his presentation. So more on this later in Per's presentation. And the other one was, how data intensive are the indicators? So we can, Sean has proposed in his presentation different ways of obtaining data to calculate these indicators. We have, importantly perhaps to say, that indicators 1 and 2 do not strictly need genetic data in the sense of DNA analysis data. It will be not always sufficient, but nevertheless already very helpful to have demographic data. Indicator 1 will be on the effective size of populations. So there I refer to Linda's talk, who linked the effective size to the census size or to different demographic aspects of populations. indicator 2 is on the number of, or the proportion of populations maintained. So there, the data on the geographic distribution of species will be sufficient. And indicator 3, it is an indicator on the effort of genetic monitoring that is being done at the country level. So there you will look into what is the number or proportion of species for which you have DNA analysis data. That will probably be a tricky indicator for countries that have not much resources into DNA monitoring at this level. But it is important, as has been stated before, to have ambitious targets and indicators that measure our progress towards them. So this is an indicator that will perhaps be in the low numbers now, but we hope that many countries with these initiatives on the importance of genetic diversity will get incentives to start monitoring and also using DNA tools for doing so.
Thank you, Miriam, so much for addressing the question. And we also received really important questions, probably one of the most important from Sung-Ming Hao, who asked, how can we let the negotiators understand scientific indicators more easily? Most of them are not scientists, but policymakers or lawyers. So Gernot posted a comment, so maybe Gernot, you would like to comment some more.
Microphone, Gernot.
Sorry, thanks, Miriam. I think this is a very important point, especially that there's still kind of some resistance on genetic indicators. However, we try to do our best in putting a number of policy briefs together, really highlighting on a very basic level why this is important and which are available for distribution, and you might be able to use for introducing the concepts to people not familiar with from a scientific point of level. And obviously, if you have any more specific questions on that, then we are always there to help you in explaining that.
Perhaps to add that really there is materials, and we are trying to make this easier by publishing policy briefs every time a scientific paper comes out, and keep these policy briefs in simple language so that they are the most accessible possible really for policymakers.
Thank you. Another question came.
Sorry, Dharma.
Yes.
Probably Uma would like to comment on this.
Sorry, just a very small comment, but I think it might be nice to also include other examples which are local, because this might sometimes allow the communication process better than other examples. So maybe that's something which we can think about in the context of Asia, is examples from Asia. Thank you.
Or any region as it matters, yeah? Yeah, absolutely. It helps to relate easier to the example, yes. Wonderful comment. Thank you so much. We had a question from Mrs. Juliana Susanti, who asked, is it necessary to consider different type of ecosystem to measure the trend of genetic diversity of wildlife species?
Yes, I mean, we think that's very important, and we envision a process in getting our indicators into work. So we don't think that we can use them on everything, of course. Rather, we envision that some key species are at focus at national level. So perhaps 50 species or 100 species, a set number that represent different kinds of ecosystems and different kind of ecosystem functions. So that's our envision for the indicators. And also we hope that relating to the question on how to make it easier for managers and negotiators to understand, I mean, a key thing is to understand that genetic diversity is really important, that it's at the DNA level where evolution occurs. If we don't have this diversity, we have nothing else. So I think that is something that needs to be communicated, and that all of us who are involved in some respect into the CBD works need to communicate over and over again. And then our indicators now are simplified in the way that indicators 1 and 2 don't necessarily need genetic data. Rather, we can use sensor size as a proxy. And that seems to be, that we think is hopefully easy enough. You need a large population to maintain genetic diversity. And we have this focal value of 5,000 that represents effective size of 500 approximately. Thanks.
Thank you, Dr. Leikne. Anyone else maybe to add?
Yeah, Emma, I would like to reply immediately to Santiago Gonzalez, who raised a very interesting point about our indicator number 3.
Yes.
And of course, it's said in the forestry's domain, you can access a level of reproductive output and natural regeneration very easily, relatively easily. And this can be approximated to a genetic monitoring exercise. And I completely agree with him. When we say that this indicator is related to the current genetic monitoring program, we are not pretending to consider this program just related to a molecular marker, something like that. As long as we manage to capture the genetic diversity and they manage to demonstrate that this program are running since years, and so they can inform us about how genetic diversity is lost or maintained or even increased, I think that's fair enough. Of course, we are putting the case with DNA markers, because for some species we know, but especially for large carnivores, it's since several years, but DNA-based methods are being used for this kind of monitoring. But of course, we are not restricting ourselves. Here, it's a concept like Linda tried to explain. Of course, we are not pretending our indicator to be the best for all the different domains of life. But since we are required to be, as long as possible, universal, we need to define something that can be applied everywhere. Of course, in some cases, you should rely on a little bit different approach, but since the aim is the same, so practically measuring genetic erosion, maintenance of a genetically different population, it's important to agree on that. Then we can discuss on different specificities. But as long as we don't agree on that, that's a problem. So that's the reason why our proposal has not to be considered, mandatory. And of course, we can still amend a bit, but it's important to acknowledge the importance of measuring and monitoring genetic diversity. So I hope to have a reply more or less to Santi. So Santi, otherwise, just let us know.
Thank you, Dr. Agnesi. So we have a comment. Just to make sure, is there also guidelines to identify the key species as you explained previously? I assume that in the case of Indonesia, as an archipelagic country, it would be a big challenge to decide which species become a key species.
So not yet. So that's one thing that needs to be developed as we start to use these indicators. So we are right now we are continuing work on these indicators. And one of the things we need to come up with is guidelines for how to put them into practice. So that remains to be done. So not yet, but soon we will have it, we hope.
Yes, probably on top of that, I would like to add to Linda's comment the following. To set up guidelines, to set up how to use these indicators, we really need your help and the help of CBD delegates. So we know that already some countries express the interest to help us in defining how to install these indicators into a practical framework. So feel free really to make your statement in favor, or also why not in this favor, explaining why to this indicator, because it's so important for us. I mean, as a scientist, we have no, I would say, we have no voice within the CBD, organization for proposing and advancing the indicators. You are the main players here. Of course, we are here for giving you what we know, but of course, we are here also for receiving what probably we don't know about the difficulties of implementing new proposal in the CBD Secretariat. And so really feel free to raise your hand and to say, look, I would be interested in finding out how to translate what we just explained into something practical for my country. Because you know that CBD process is a country-based process. So it's so important that a single country would accept to be, to some extent, a champion of these indicators, to propose also to other countries the examples of how these indicators might be used in a practical setting.
Thank you, Dr. Venezi, so much for your comment. So if we don't have further comments or questions right now, I propose to move to the final presentation because we will have additional time for discussion afterwards. So our final presenter today is Dr. Per Sjørgen Gulve, who is a specialist in population viability analysis. He is an associate professor in conservation biology at Uppsala University, and he is a senior scientific advisor at the Swedish Environmental Protection Agency. He will give talk titled Applying Proposed Indicators to CBD, the Practice. So Professor, please proceed with your presentation.
Thank you very much. If I can. Okay, can you see this?
Yes, would be nice if you can.
Also?
Yeah, but it would be nice if you put in. Okay, that's fine. Perfect, Per. Please.
Okay, so I work at the Swedish Environmental Protection Agency. I don't know how many of you know my colleague Johan Abenius, who is the Swedish national focal point for the substa of the CBD. But I'm in the same agency as he. So I was having a look at how can this be kind of, to illustrate the procedure, what do we have in Sweden and how can we attempt to use these indicators? And I will focus on two of them, and that is the effective size indicator and then the proportion of populations maintained. And I will do this basically by using data from the red list assessments in Sweden. So this is at the national level for the most part. And if you look at what data is available, it's really a combination of scientific and indigenous knowledge. So for instance, we have Sami people to the right that are actually helping in reporting to us where there are large carnivores, for instance, and together with people from the regional conservation authorities, they cooperate so that there is more knowledge about where species are present, things like that. And also dialogue with people and getting indigenous knowledge, and the cross-fertilization between scientific knowledge and indigenous knowledge is extremely helpful in advancing the knowledge on biodiversity throughout Sweden and throughout the Nordic countries, I would say, in Northern Europe. Another very important aspect is, of course, the international cooperation. So Sweden has learned a lot from scientists and experts in other countries, and of course also from people with knowledge in indigenous knowledge. So the international cooperation is really important, and this is of course a natural part of the scientific work throughout the world, and it has several win-wins, I would say, in society. So in Sweden it has benefited, for instance, education to a great extent, and the knowledge on various aspects of utility and the good thing about biodiversity. much of that is actually being collected from cooperation between people in Sweden and people in other countries, and we are learning from each other, and it's really also benefiting our education system greatly. And another one is, of course, that it also creates jobs, that we are investing a lot of resources also to develop things, but also with this comes a win-win, that people are doing things voluntarily, for instance, citizen science, which leads to additional reporting and monitoring. And so what we have done from the Swedish EPA, my agency, is that we have invested in a web-based portal where you can actually enter your observations on various species. And I will just show you a little bit about this. But of course, in order to store information about observations of individuals of species or of species, this helps, of course, to see where they are present and they seem to be absent. But it also requires, of course, a national, or it could be used maybe an international database for this. Of course, and it's also a combination of how do you census. So there are ecological census and estimation methods to do this in a more scientific way for the red list assessment, for instance. But there are also, of course, expert judgments and things like that. And there's also, of course, the genetic method that you can actually get an estimate of effective population size from, for instance, repeat. Sorry?
Go ahead, Per.
Can we take the question maybe afterwards?
Yes, absolutely.
Okay, right. And so another important aspect of the genetic methods is, of course, to use genetic markers to examine is there connectivity between subpopulations of the same species in different parts of landscapes or parts of the country. Another aspect is, of course, also to do a genetic investigation to assess the distinctness, the taxonomic status of a certain population of a species. What we see here and what we also work with in the red listing procedure is, of course, a combination of detailed checkpoints where you do a census of the species and the populations, and then that you have some kind of combined trend assessment that you are using a methodology for, like in the WWF Living Planet Index. So my examples come from Northern Europe, and it's about Sweden and Fennoscandia. And Fennoscandia is then Sweden, Norway, and Finland together. Finland and Norway are situated on this peninsula from north to south, and Finland is on the mainland, you could say. And Finland and Sweden are member states of the European Union, and that means that we are doing reporting of certain species and habitats in a kind of standardized fashion decided together within the European Union. And that is called also the Natura 2000 that you might have heard of. And all these three countries, they have ratified, for instance, the Berne Convention on the protection of species and habitats throughout Europe. And they are also have ratified the Convention on Biological Diversity. And these things makes things easier to cooperate, of course, also. But also for our part, the IUCN Red List and also the EU species status assessments, they help a lot, so that we are kind of, to a greater extent, harmonizing how we work, as would be the case probably if these kind of cooperations did not occur at all. One special thing here is that both Norway and Sweden has national species information centers. And of course, this is an investment from the federal level, but it has helped a lot, and it's been very fruitful also for education of the public and also of people that work out with forestry or in the agriculture, for instance. So it's been very beneficial. And also, of course, the databases for storing species records and observations, of course, this comes with this investment. In Sweden, genetic monitoring is so far quite unusual, but it is likely to be used much more, and therefore I cannot say very much about the indicator 3 in the Hoban et al. Paper. So I will get into more of the indicators 1 and 2. And I've used data just in a handful of case studies from the Swedish species portal. You can see the URL, or the, what do you say, the web address there to the left. And here's the first species, and this is just a handful of cases to illustrate. My focal thing here is also that here we have two distinct meta populations of this butterfly, the butterfly Lopinga akene, which is a near threatened species in Sweden. So one meta population system is out on an island in the Baltic, and the other one is on the mainland. And from the data we have, we can then use this conservative ratio between the genetic effective size and the census size, and the census size is then the number of estimated number of adults in the system. And so for this 0.1 rule, the census size for 500 effective size genetically, the corresponding census size would be 5,000 individuals for the meta population, for instance, where you have interconnected local populations. And what were the observations then? Well, in the last red listing, there were more than 6,100 butterflies estimated in the mainland population, meta population. And on the island Gotland, there were around 7,000, that is the estimate there. So from this, we can conclude that both these meta populations, they still exist and they seem to be stable. There is a recovery program to kind of give the good management of the habitats they live in. And so both these meta populations remain. Here's another example, which is the large blue butterfly, and it is also classified in the red list as near threatened. And with this conservative 0.1 rule, the census size would be 5,000 once again for a distinct population. And here you can see that it's not as distinct as the previous case. You see that there are two islands that have quite a lot of them, and then there are kind of like scattered occurrences in southern Sweden. And it also turns out that some of these scattered occurrences, they have disappeared, so they have gone extinct. But not, the species has not gone extinct nationally in Sweden. And so from the meta population sizes, we can see here that on these two islands, there are actually good populations. So they are in the order of 5,000 individuals. But on the mainland, they are smaller, and also some of them have disappeared. So I will get back to this. But if you look at the proportion of distinct populations that remain, it's approximately 77%. And the species as present is decreasing, but there is a recovery program existing. And there's a third case example, and that is the pool frog. And with the conservative 0.1 rule, the census size again would be 5,000. And it occurs as one big meta population along the coast in mid-Sweden, but then there's also a natural small population occurring in the southern part. And if you look at the estimates from how many frogs, you can see that they are in the order of 5,000 total in Sweden, and they are subdivided into around 100 in this separate local two populations, and then 4,900 in the larger meta population. But none of these are big enough to be corresponding to effective population size that is larger than 500. But all these two remains, and there is a recovery program. And here is a example with a large carnivore that is transnational, so that it has actually gene flow between subpopulations that occur in all three countries. So it's a transboundary population, and it's a large carnivore. And from this 0.1 rule, the census size again would be then 5,000 in order to be effective size 500. But here we have done studies. So from both simulation studies with detailed modeling and also from genetic studies, it's actually known that the ratio between effective size and census size is in the order of 25%. So it means that the required census size at this level among these three countries to have a population that is larger than 500 genetic effective size, it would require more than 2,000 individuals. Okay, but so how many were there? Well, in the latest census in all three countries, which was the previous year, it turned out that there were almost 1,400 wolverines in total. And so that is much lower than the 2018. That would be the kind of minimum in order to have the effective size of 500. So for this species, it's not among those that have an effective size larger than 500. But 100% of the populations remain, and it's increasing actually, and management plans exist. And what we are doing is to increase the genetic monitoring and also the co-management between countries, so that we are doing management together into a larger part than we have done before. And here's a last example just of breeds. In this case, there were 5 Swedish breeds of a sheep, Ovis aries. And with this, it was actually done a genetic study with genetic methods. And it turned out that these 5 breeds, they had very low effective sizes genetically. So if we say then 500 genetic effective size, it turned out that these breeds had 16 or were 32, 38, 68, and the breed with the largest effective population size, according to these methods, were in the order of 81. So here there needs to be very much more done in order to increase the effective size of each of these breeds. But all of them remain, so there are management plans that exist. So boiling down this then to you can see here then that for the Lupinga butterfly that I started off with, we had 2 genetically distinct populations, and both of them were larger in census size that would say that they are larger than 500 effective size. And for the second butterfly, there was a mixture, so that there were 2 populations that were big enough, and there were 3 populations that were not big enough to order to qualify for the effective size 500. For the Maclinea butterfly, there was also a loss of populations. So you can see it there. For the frog, the Pelophylax, there were 2 populations, but none of them qualified for having larger than 500 effective size. And the same also then for the Gula wolverine, but it was just 1 transboundary population. And with our, if you take an average of this, then the remaining distinct populations here for the country, just using these 4 examples, would be 94% averaged over the species. Another question that has come up when we've discussed this is, what about species that have gone extinct? Are they kind of vanishing from this? And we think that if there are species that have gone regionally extinct in a country or that have gone globally extinct, it's not that they disappear, but they have to be also among the species that have less than 500 effective size, so that you're kind of not benefiting from species going extinct. And also probably according to the IUCN criteria for population size for critically endangered species, it's quite certain that they will not have effective population size larger than 500. And so they can also probably be included in this listing per country if there are such assessments as a species that has not effective size larger than 500. And for the Swedish breeds, the 5 of them, the genetic indicator then for effective size was 0 because none of them had large enough effective size. And the remaining distinct populations among them, the remaining breeds were 100%. So this was a case with not very much genetic data, but I think that might be a real situation for many other countries too. So, and just a handful examples. So thank you very much.
Thank you very much for your presentation. And now we again have some time for discussions. In the meantime, that we don't receive, until we receive any further questions, do you want maybe to comment further on the previous question regarding the species selection with a monitoring program? Gernot and Joachim posted some comments, and maybe it would be a good place to do it in person now. And especially after we heard all 3 presentations.
Yes, I can just say for Sweden that we have found the proposed indicators of Hoban et al. Extremely interesting. And so we are, at our own expense, of course, then commissioning a test. So we have a species information center, and we are discussing with them now, asking them to test these indicators to see, for instance, for which organism groups does it seem that they are more easily applied, and for which organism groups do they judge that it's impossible or whatever, just to get some feedback on where could this be done, and would there be methods that would be very good in to do this in a resource effective way. But we take it very seriously that it's also very important to conserve the genetic diversity, and not only among species associated with agriculture like breeds and sorts, but also for wild species, because they also are supplying a lot of ecosystem services, but they are also very important to kind of have the ecosystems as intact as possible, and to be able to withstand climate change, for instance.
Yes, this is a further comment on the previous discussion regarding the isolated population and islands. So Mrs. Yap commented, for small island states like Singapore, we may have the diversity, but not the population size that these indicators require.
Yes, and there might be like the wolverine, the large carnivore example that I had, that with instance, with cooperation with Malaysia, that maybe you are you have the possibility to do assessments together, that there are species that you share. That might be, I was thinking of birds, for instance, that it might be that birds, some do not migrate, of course, and disperse, but many might do, and so that you can do maybe assessments together. Yes, getting data. Of course, I know that there are some initiatives on citizen science on bird reporting of birds, for instance, and maybe there could be similar initiatives also for other taxa. organism groups that could help in this. Yes, Uma.
Yeah, sorry, I just wanted to point out, because you mentioned citizen science, there's a recent report called The State of India's Birds, where over 10 million citizen science records were used to try and look at trends in certain bird species. And I think that it's a very powerful tool in Asia. I'll just post the link here. But I think such approaches, because there's lots of people who are interested in biodiversity. And I think it's a really great way to involve public and also gather information. Thank you.
Yes. And also I would encourage very much the international collaboration also, and I mean also between people that are members of non-governmental organizations and scientists that are really interested also. And also for these census data, it's not the same as genetic data with sequences of the DNA and things like that. So it's a bit easier maybe to share data with respect to monitoring and just counts of and estimates of populations that way.
Yes, Joachim?
Yeah, I want to comment further on the question from, or the comment on from Wendy Yap for the small island states like Singapore. It is of course important to consider that organisms don't have political boundaries. So this requires to look at the level of the population. For some species, you will have populations within Singapore. For other species, it will be transboundary populations with Malaysia, for example. So this requires this international collaboration to see at what level these populations are actually evaluated. And there are some examples that Per also gave. So for the wolverine, that they're not evaluating this on the level of the different countries itself, but on the level of the northern Fennoscandia for this species. And depending on the species, this is an exercise that needs to be done over and over again.
Thank you, Joachim. And there was a mentioning of the data sharing. Do you think, is there a need maybe to discuss a bit about possible problems regarding this aspect in terms of the regional collaboration?
I think that is something that could be very also useful from the IUCN, for instance, the World Conservation Monitoring Center, to well, discuss with them how they have arranged and look at, for instance, if there's similar collaborations. I was thinking also of the WWF Living Planet Index that has been applied to birds and mammals primarily, but could probably be developed also for other organism groups to a larger extent. And then, of course, also if it comes to genetic data, it might be that we are storing kind of like measurements or indicators that are not the basic gene codes, but are something else, and that will not be as how do you say, controversial maybe with respect to handling of genetic information.
Uma, please go ahead.
Yeah, this is just an idea and probably will take a lot of effort, but it's also maybe possible to think about databases where data from a country is private. So like but the database can be queried. So for example, these would be very useful in something very applied like forensics, but also generally if you have cross-boundary species, like say the snow leopard or something, right? So if each country could upload their data and somehow I couldn't see data from other countries, but I could query the database or some such thing. Some ways to have data privacy, but also data sharing might be a really good way for the future. Thank you.
Yes, and also I think that if you do, for instance, like genetic measurements using genetic methods, that this is something you do within your country, so that it isn't that you're sharing the raw data, but that you're communicating the results more or less, that there are some measurements that are indicators more or less, and not the kind of basic codes. Linda, your mic.
Yes, I just wanted to relate to Wendy Japp's comments that for some situations, populations may not reach this target of 5,000 or the NE 500. And of course, that would be the case. And I think you highlighted that, Per, that we have such situations in Sweden also where we don't meet these goals. But I think that's important to realize, that we in many cases have too small populations to be able to effectively conserve genetic diversity. And we need to become aware of that and then try to take measures to change it. So, I mean, for the wolverine, we hope that international collaboration can result in a larger meta-population for this species, and the same thing for the breeds and the other populations that you mentioned. So that will be a conservation aim that we hope the indicators can highlight, that we need to increase population size in several cases. Thanks.
Yes, and in the case of the wolverine, Norway and Sweden has in the order of 1,000 wolverines together, but then Finland just has well, less than 400. So it's quite a lot up to them to increase their population size, and that might be one way to get a larger population size for the transboundary population.
I wanted also to highlight that international collaboration in genetic data has other benefits. And you may look up the annex of the Nagoya Protocol, where you have basically all non-monetary benefits that go from increased capacity building, increased product development that may be possible on some plants, for example, training, even raising awareness that can really help in biodiversity conservation in the long run. So it's a matter of liaising with the relevant partners and come up with transnational initiatives that have a lot of indirect benefits. So I will put up this list of non-monetary benefits of the Nagoya Protocol together with the webinar documents, so you have a possibility to consult them, hopefully to convince you that international collaboration is a good thing in this initiative of biodiversity conservation.
Thank you, Miriam, so much for your comments. I propose to keep up with the schedule. I know that all of us have other obligations today, so I thank you so much all for participating. And to our respected CBD representatives and national focal points, we kindly thank you for your participation today. We hope that you found our webinar beneficial and that our conversation will not end with it, but only begin. So please feel free to send us your questions, comments, concerns, doubts through email. We will make sure to send you all the materials from today's webinar and of course to provide you with any additional information that you need regarding this topic. As your feedback is really important to us, we will also send you a questionnaire regarding today's webinar that we really ask you to fill in and send us back in order to help us improve our efforts. So for the closing, I would ask Dr. Veronesi to give us his closing words. So Dr. Veronesi, please.
Yes, I can only say really thanks to all of you for your patience, for attending this 2 hours webinar. And just coming back to, I think it was from Singapore, this comment, we like to build a kind of coalition in which the capacity building aspect will be probably the main focus. What does it mean? It means that we are going to probably create a network of researchers willing to provide significant help to people requiring specific topics to be addressed on a scientific basis regarding these genetic indicators. So as Belma pointed out, this is just the beginning, and we really hope that you can follow us in this process, but it's going to take quite a long time. We know that the CBD is working hard on this. You got a lot of meetings, so please feel free to ask any of us comments, specific help. We are here for really helping you and make this path together towards the inclusion of genetic indicators into the new CBD policy. So once again, thanks to all of you. Have a nice day and hope to see you soon in a new webinar. Bye to everyone.
Thank you. Bye.